Well, hello everyone, it's Chow here. So I've been asked by a number of people as to how to approach this question that I posted on the review slides. And I think that I'll just go ahead and make this quick video showing everyone basically how to read the genetic code table as well as how to figure out this sequence. It's actually not too difficult, but there is sort of a little bit of trickiness to it. But I think we can just go through this, and hopefully by the end of it you'll have a better idea as to how to get a protein sequence, or I guess I should say an amino acid polypeptide sequence, from an mRNA. So let's get started. So this is the strand that I basically put out for everyone, and it's a bunch of U's and A's and G's and C's, and it looks kind of scary, but I did put it in the 5' to 3' direction. Again, remember that when you're reading the strand with a ribosome, you are going from the 5' to the 3' direction. So now you're giving a bunch of choices, but honestly these choices don't really even matter because the important part is learning how to figure out this table over here to the right. So the first thing you should do is actually find the start codon. So if you remember, the start codon is always AUG, at least generally speaking. So you find the start codon, you just scan through this thing and you go, oh, there's the AUG, so there's your start codon. So after that, what you want to do is start figuring out the following codons. So one thing you can do to basically help you figure out the following codons is actually breaking the sequence up. So you have the initial start codon, and remember, codons are 3 RNA bases, so just go ahead and break apart your mRNA strand over here. And then you should find the stop codon in the sequence. So remember, stop codon is UAA, UAG, and UGA. So here in this instance, it's UGA. That's the stop codon that I specified. So you find your stop codon. Pretty awesome, yeah? So that's pretty straightforward because now you can just go ahead and use this table to help you figure everything out. So the first thing you should do is learn how to read this table. Now, I'm not sure if on the exam you'll be asked this exact question per se, but it is a good concept to understand. So how you approach reading this table is basically by going from the left side to the top and then to the right side. So let's look at this sequence over here. Now AUG, you should know that that's methionine, but let's say you don't know that's methionine. What you want to do is go over to this genetic code table over here, or also known as an RNA codon table, and you look at the first letter, which is always on the left side, and the first letter here is A, so you go to A, which is over here. The second letter is U, so you want to go to the U's, which again, second letter is at the top, so you want to go to the U's. And then finally, you're in this section over here. The last letter is G, so you want to go find A, U, and then over here to the right, you want to look for G, and there you go, AUG, methionine start codon. Next, you move on to the second one. Here I specified CAA. So you go over here, C, and then you move over to the A section over here at the top, so it's in here, and then you look at A in the third section, so the right over here, and you find that CAA is glutamine. So you have methionine for the first one, the AUG, and now you have glutamine. And then next we have ACU, so again A over here, and then we go to C over here, so it's in this box, and then finally you go find U in the right side, and ACU, that's threonine. And then you do the same for the following codons all the way until you get to the stop codon, which again, stop codons don't actually code for any specific amino acid, but it does cause a release factor to come in and basically tell the whole complex to stop translation. So you have this sequence and you go on and on and on until you reach a stop codon, and therefore you should get a final sequence. And in this example, the correct answer is D, because, well, if you go through everything from the AUG all the way to the, let's see, that's AGU, that should be this sequence over here. So this is how you approach a situation where you have a table and you have an mRNA strand. Another thing that can be potentially helpful is actually working backwards. So having this particular sequence over here and then trying to get an RNA or possibly even a DNA sequence using your knowledge. But of course, you have to keep in mind that sometimes, like I said before, you can have instances where different codons code for the same amino acid, so you might not get the 100% always accurate strand. But it is a way to practice. So I hope everyone found this short video useful, and I will see you later. And good luck studying, everyone.